From Microbe TV, this is TWiN, This Week in Neuroscience, episode 32, recorded on July 25th, 2022. I'm Ori Lieberman, and you're listening to the podcast about the nervous system. So welcome in, everybody. I'm taking over for Vincent, who is at a BAT meeting, which sounds perhaps more fun than what we are doing here. But let me introduce who we have with us. So from New York University, we have Timothy Chung.
Hello. Hey, Ori. Hi, everyone. Good to be back.
I don't think I've ever called you Timothy before, but it's always a first.
Oh, I wasn't even listening.
We're getting super formal.
And then from Vanderbilt still?
Oh, no,
that's old.
It's been updated.
I'm on old from Tulane University, Vivian Morrison.
Hello. Good to be back.
Hello. So if you are watching on YouTube, you will notice that I have shaved my head, which is merely for ease of use in the setting of having a child.
How does that? Can you explain that? Why did you shave your head but not your beard?
Well, I feel like the showers are very infrequent, so it looks less gross, and also I felt like this is where my balding pattern was headed, and I'm off work for a while, so I forgot about the video.
But yeah, if you were to also shave your beard, then you would look like your baby.
Exactly. No, I have one picture of the two of us where we look exactly the same. Maybe we'll post it in the show notes.
Well, or you could just put some, make him have a beard.
He does have some serious baby acne, so.
Oh no,
it's okay. It's it's cute.
Hopefully he'll get it out of the way and not have to deal with it during puberty.
No, then I was like, maybe there's a correlation. I don't know. That was on my list of things to look at PubMed about. How are you guys doing?
Doing great. It's going crazy outside weather-wise. So yeah, it's I don't know whether the thundering is going to leak through the microphone. It'll be exciting.
Yeah,
I'm sure it's hot in New Orleans. Yeah.
Yeah, it's.
Is it hotter than Switzerland? Wait, it's as hot in Switzerland as here?
Not as much a differential.
Oh, wow.
There's not as much of a differential. You would think. The big difference is that it's not really humid there,
okay.
But they don't have air conditioning. So
no, they don't. Europe is heating up as a whole. Mm-hmm. London was up at 40 Celsius last week. It's absolutely crazy. Yeah.
If you look at the map of the U.S. with temperatures, though, then there's San Francisco, which is a cool 65 every day. So guys are welcome here. Yeah. Okay, so today we are going to talk about an interesting paper that kind of came across my radar from looking at basic science mechanisms of clinical research. And although we will talk about the paper for the most part, I think we'll kind of take a step back and talk a little bit about something that we haven't talked yet about on TWiN before, which is stroke. So first, let me mention the paper, which is titled Coordinated Increase of Reliable Cortical and Striatal Ensemble Activations During Recovery After Stroke. It was published in Cell Reports in 2021. The first author is Ling Guo, and I think the co-first author is Shravani Kondapavulur, who is actually a UCSF MD PhD student, and I have interacted with her a couple times. And then the last author is Karunesh Ganguly, who is just a prolific scientist studying motor learning and kind of, I think, going towards an idea of maybe brain-machine interfaces for motor recovery after stroke. So maybe we can start, and I think we'll do two pieces of background here, and one of which I'll rely more heavily on you guys for, which is, so the first we'll talk about stroke in general, we'll talk about recovery after stroke, then we'll talk about motor learning, and I know that Tim has a kind of a big, he's making a face, but has done a lot of work on this. And then we'll talk about this paper, which is heavy on the math, which we can kind of glaze over.
That was sarcasm when you said that earlier, Tim.
Oh, that was complete sarcasm. This paper is terrifying.
All right, great.
Yeah, we can go in as much as we want, hopefully led by Tim, and then
Oh no.
we'll kind of wrap up. Um, so so what is a stroke? So I think that the public messaging on stroke is really strong. There's okay, if you see a facial droop or your speech gets slurred or you're weak, you should go to the hospital right away because time is brain. Um, and the way we think about it as neurologists and neuroscientists is that stroke is a sudden onset deficit in brain function that happens from a vascular event. So that could be either bleeding in the brain or a clot in the brain. And that leads to decreased oxygenation and perfusion of brain tissue and then dysfunction of the brain tissue itself. And
Actually,
yeah,
quick
go ahead.
question, Ori. Ori, you're a neurologist, and I thought I heard news that COVID can cause blood clot and weird vascular problems. So I'm wondering if there have been upticks of stroke patients during COVID.
There were at the beginning, or there were strokes occurring in populations that don't normally get impacted by stroke.
Yeah, I think in young people, I feel like it's often invoked. I don't know what the data are for it, but on the other hand, like COVID is so prevalent that if someone comes in with a stroke and they have COVID, it's like, okay, do they just have COVID?
No.
Yeah. But certainly there's increased risk for, there's like kind of like a thrombophilia and increased risk of clotting in COVID. And there have been a lot of trials aimed at thinking about like using blood thinners prophylactically to prevent clots in patients that are very sick with COVID. And some of those trials have been positive. But there certainly, I think, is some signal that there is an association between COVID and stroke in young people.
Although what are your guys' thoughts on how that might be changing as the virus changes? Because now it's become more like a cold virus. Just I haven't heard as much about those kind of strange adverse events.
Yeah, I guess I don't know. I think that as we're kind of learning in real time, but there's always people who are coming in who have bad vasculature and are good, maybe have a stroke anyway, and then COVID just sets them off their equilibrium or pushes them a little bit towards that. The question that I always have is like, is that any different from any other viral infection that could cause that? And I don't know it's pretty hard to test that. And certainly like as the cases get more mild, as we have more vaccines, then certainly they should be less severe COVID disease and maybe therefore less stroke or less sequelae of it. Yeah. So I think the important thing to understand about stroke is that it can affect any part of the brain. And in this case, in this paper, and kind of I think in the public's eye, stroke affects the motor system and can cause paralysis on one side of the body or weakness on one side of the body or the other. But it can, of course, cause many different things. So it can cause memory problems, it can cause sensory problems, it can cause coordination problems, and then it can also cause this thing called neglect, which I find very interesting, where even though a patient can use both arms, they will preferentially use one over the other. So lots of different kind of sequelae of having a stroke. And how does a stroke happen? So a stroke can happen from a number of different reasons. As you brought up with COVID, it can be that the blood is more coagulable, so you have more clotting. More classically, it can happen from someone with atrial fibrillation, so a weird rhythm in the heart where the heart doesn't contract as well, and a clot can form in the heart, and then it will fly to the brain. You can also have atherosclerosis, so like plaques building up in blood vessels that can kind of have a clot form there and then go to the brain, or it could even happen in the brain's vasculature itself. And then you can have small strokes that are associated with hypertension or hyperlipidemia, so high cholesterol or diabetes. And all of these have different patterns of kind of deficits and all of this stuff. And I think that clinically, those are kind of the questions that we try to answer because we want to prevent stroke from happening again. But let's say someone has a stroke, what happens to them? So I think maybe even before I became a neurology resident, I thought that, okay, someone has a stroke and their deficits are fixed, that this part of the brain is infarcted, has died, and you're never going to recover brain function. But what's amazing is that that's actually not the case. So there's a lot of recovery after stroke and plasticity in the brain. So that, for example, within 3 months, many people can have large improvements in their neurologic deficits after the stroke. And there's been a lot of research that is focused on trying to understand the underlying mechanisms and also brain networks that control this recovery. Because as you can imagine, enhancing recovery would be a great way to improve the lives of patients with stroke. And this paper kind of takes off from that by saying, okay, if we give a bunch of rats strokes, and we'll talk about kind of the exact way they did this in a second, what happens to the brain? How does the activity of the brain change? And does it support the motor recovery, or how does that activity change support the motor recovery in these rats after the stroke? So tell me what you guys think about that, or do you think I missed anything, or kind of how does that set up for the paper?
Sounds good to me. I didn't know rats can have strokes.
Well, you didn't?
I don't think rats
they don't have it on their own.
Yeah, I think we can make rats have strokes.
They might in the wild. I don't know.
So let's go ahead, Vivian.
Well, I guess I don't know if you're going to touch on this, Ori, when you talk about the method that they use, but I want to flag that I think there's controversy in the stroke field about whether or not you can take the results that are found with one stroke model and assume that they're going to apply in a different stroke model. So I think, just the results of this paper may only be applicable or largely applicable in the context of this model, but they might not be as appropriate for a different model.
Yeah. So I think maybe we can kind of talk about the motor system, and then we can go back to thinking about the exact model, because I think that's a really good point. So Tim, let's talk a little bit about the kind of the pyramidal and the extrapyramidal motor systems. So I'll talk about the pyramidal one, then I'll hand it off. So the pyramidal motor system, so is kind of the classic way many people think about movement. So it starts in the motor cortex, and there are axons that travel down through different parts of the brain into the spinal cord, and then synapse, and travel out into the peripheral nerves that control muscle contraction. And the motor cortex itself was kind of studied by this guy named Penfield, who classically stimulated different parts of the motor cortex. And if you stimulated one part, the person's arm would move, and if you stimulated another part, their leg would move, and another part, their face would wince. And so the idea is that there's kind of a one-to-one correlation between motor cortex and controlling different large areas of a limb or a part of the body in terms of movement. And that
the homunculus.
The
homunculus.
Yeah. And that is important for both kind of having large order gross movements, so flinging an arm, but also fine movements and learning new movements as well. So for example, buttoning a shirt or chopping a knife, or I should have thought of other examples of this before I said that. But that learning happens in the motor cortex and in other parts of the brain. And then there's a separate part of kind of the motor system that's called the extrapyramidal system, and where the motor cortex sends information to kind of internal loops within the brain that then goes back to the motor cortex before, so it can essentially modify itself based on other parameters, such as motivation or internal state. And maybe Tim can tell us a little bit more about that.
I'll try. So as Ori mentioned, the pyramidal system is going from your motor cortex down to your spine, and then controls the movement of your hands and stuff. But those neurons also kind of bifurcate in a way. They send so-called collaterals. They send kind of side branches into the striatum, which is part of your brain that is critical also for movement. And And you know that's true because in patients that have Parkinson's disease, they lose the dopamine system, the dopamine innervation that talks to the striatum. And when that happens, then the patients cannot move anymore, even though we think largely the cortex, the neurons are still there and they are still able to fire. And the only problem is that this kind of side branch in the striatum is gumming up the works. And the way the striatum works is the striatum receives input from the cortex, and the striatum kind of sends axons down to other bits of the basal ganglia. It is the bit of the brain that is at the, as you can tell from the name, it is a bunch of brain nuclei that is at the bottom of the brain, more bottom relative to the cortex. And it sends a loop all the way to the thalamus, which is classically thought of as the relay station of the brain. And through the thalamus, it talks back to the motor cortex. And I think neuroscientists largely think that it is through this loop, so from the motor cortex to the basal ganglia, including the striatum, to the thalamus, back to the motor cortex, that allows some of the learning to be accomplished. And also, not just learning in terms of how you move your hands and stuff, but also abnormal learning, such as perhaps drug addiction, which also engages the dopamine system and the basal ganglia.
Yes. I'll just add that I think like there's this prevailing view now, which I don't think I feel like I didn't learn about in neuroscience courses, but that the basal ganglia control the vigor of movements. So, for example, you could learn how to button a shirt, but if you're not motivated to button the shirt, or then you're not going to button it as robustly and vigorously or quickly or as efficiently, and that information is given to the motor system by the basal ganglia, which is a long-winded way of saying that it doesn't just convey the act and kind of like the program of what to do with your hands, but it's how you use that motor program to accomplish a task.
Does anybody know do either of you know about connectivity between the networks and structures that you're talking about and the cerebellum? Which some of the things you mentioned, I'm like, I typically have attributed that to the cerebellum, like being able to time. You said like, how efficient are you at doing something? So timing movements, putting them together in in a string that makes sense, and then also the kind of like intensity of the movement.
Yeah, I guess I think the way that I think about the cerebellum is that that like the cerebellum is like an online like modifier of ongoing action. So it says, based on the sensory information, are you doing this action correctly while it's happening? And then how can you coordinate other muscle groups or other parts of the body to achieve like complete the motor program? Mm-hmm. But I think there's always more things that are coming out about the cerebellum, like having dopamine innervation and kind of incorporating more cognitive functions as it comes to motor activity. But I think classically, I think about it more as like coordination, which is less so about the sequence of events or kind of how vigorously you do them, but more about how you do them effectively and kind of incorporating all the different pieces to the motor action.
Yeah, I think cerebellum is another structure that is heavily involved in motor learning, which I know essentially nothing about. The one thing that I know is that often when you move, your timing of when you do things are very important. And it's well known that cerebellum is very important for having a proper sense of timing, but only for times that are very, very short in duration. So like down to the second, millisecond and second scale of your movements. Whereas the striatum is also involved in timing, but it's for a bit of a longer kind of time. So on the seconds to tens of seconds range. So that might map to different aspects of movement. So cerebellum might be, as already said, like fine movement adjustment and stuff, like with instant proprioceptive feedback. Like, when I'm reaching for a cup of coffee, are my fingers touching the right place of the cup so I don't drop it? Whereas for the striatum, it might be more longer sequences of movement, such as, I can't, when I go eat lunch, am I going to have a good meal? That kind of stuff.
So let's talk about the model that they use in this paper, which I think, as Vivian mentioned, is kind of is obviously a one model, and it's hard to extrapolate. But so strokes can happen anywhere in the brain, right? So in humans, they can happen in the motor cortex. Certainly, they can happen and affect many other parts of cortex in addition to motor cortex. So you can have sensory deficits, you can have neglect in addition to the motor deficit. They can also happen at different parts of the pyramidal system. So as the axons leave the motor cortex, they go through kind of fiber tracks that track through other parts of the brain. And those there can be strokes there that can lead to deficits in motor function. You can also have deficits in the brain, or strokes in the brainstem, and then also ischemia or infarcts in the spinal cord, although we classically don't think about them necessarily as what we would call a stroke, right? And I think that clinically, there's a lot of heterogeneity, right? So both.
You can have stroke in the spinal cord.
Well, you can have infarct in the spinal cord, right? I don't know that I would. I don't know that that's called a stroke.
Is that? I can imagine if you have a stroke further up the spinal cord, like in your brainstem, those are dangerous because it can knock out your breathing and your consciousness. But how does stroke in the spinal cord work? Do you have local paralysis and?
Yeah, I mean, so if you have a clot that goes to one of the arteries in the spinal cord, you can either have sensory deficits, motor deficits. It can be one side or the other. There are the innervation or the vasculature that takes blood to the spinal cord is also very stereotyped, just like in the brain, essentially. But I think we wouldn't necessarily call that a stroke. We'd call that an infarct of a spinal cord infarct, right? So you can imagine where the heterogeneity in the stroke outcomes comes from, right? So if I have a stroke that affects my motor cortex, so I can't use my hand, and that same blood clot leads to brain dying in an area that leads to neglect. So then my hand's not working and I don't even regard the hand itself. It's very hard for me to learn new tasks and recover from that, right? Whereas if I just have it in the motor cortex, I can focus and pay attention to the hand and relearn and kind of regain function as the brain recovers. So the way that, unfortunately though, it's very hard to study the mechanisms of stroke recovery in a clinical population where there's lots of heterogeneity in the infarct itself, much less kind of how the brain tissue responds to that insult and has a recovery of function. So by moving into animal models, we can say, okay, what if there's just a stroke in one part of the brain? For example, in the motor cortex, how does the rest of the brain respond to that? And can we extrapolate kind of the mechanisms of recovery to mechanisms of recovery that happen if there's a stroke in another place of the brain? If you guys are all with me.
Yeah, I'm with you. But I'm just wondering, is it known which part of the brain, if you have a stroke in, it causes neglect? I'm guessing it's not the motor cortex.
No, it's usually
somewhere
in the
else.
parietal cortex, so further back.
Okay.
So there it's kind of higher order motor and sensory processing. So
where things get integrated.
Yeah, exactly. So that's super interesting. And you can have neglect in monkeys, but I'm not sure that there's a true. This is a side.
Sorry,
that's why we're here. We're here for side notes.
So I think that neglect is super interesting, and what's really cool is that neglect affects. So if you have neglect or inattention, it affects the motor recovery. And the neural mechanisms of that are not known of how that happens, okay? So, but what's really cool is if you go to animal models, the way in rodents, for example, the way you get neglect is not by lesioning parts of the cortex. I don't know if that has been shown, but the classic hemi-neglect animal model in rodents is a dopamine lesion, right? So those mice completely neglect one side of the contralateral side to the dopamine lesion. That said, that is not kind of neglect in humans is not. If you lose dopamine on one side of the brain in humans, you get Parkinson's disease, more asymmetric form of Parkinson's. So it's obviously hard to model these higher order cognitive processes in lower animals, but certainly in monkeys they can have neglect.
Okay.
in terms of kind of a motor recovery, so how, you imagine that the brain, the motor homunculus, which we talked about, is dead. How could I possibly control my hand after a stroke? And what's interesting is that areas of cortex outside of that motor, the kind of primary motor cortex, start to behave and fire as if it was motor cortex. And they will both have local plasticity and also like axon outgrowth and stuff like that. And essentially, what you see is that the function of the motor cortex then diffuses to areas of cortex around the motor cortex. And if you record from adjacent cortex, and what they call here the PLC or perilesional cortex, it actually starts to behave more like a primary motor cortex, even though the primary motor cortex itself has died in the stroke.
Presumably, importantly, in these adjacent cortex, that is not the primary motor cortex. These are like, I guess, secondary motor cortex, associative motor cortex. The neurons there still send axons down to the spine, right, to innervate the muscles.
Correct.
It's just maybe less dense axons, my guess is.
So remember, there's also like lots of intracortical connections. So the motor cortex neurons, they send axons to the spine, they send axons to the basal ganglia, to the striatum, as we talked about, but they also send axonal connections to other parts of the cortex. And so what Tim is referring to is kind of secondary motor cortex, which is involved in less in which finger should move and more in kind of planning larger scale motor actions. And I think the point is that part of the motor cortex then it essentially has the connectivity to do what the primary motor cortex does. And then it takes over that role when the primary motor cortex dies.
Right.
And that is known both in humans, looking at fMRI, and also EEG recordings, and also in animals. And work from Karunish Ganguly's group and others has shown that the perilesional cortex, or the PLC, which is really the secondary motor cortex, starts to behave as if it's the primary motor cortex. What's not known in animal models is what happens in the basal ganglia. And as you could imagine, if the primary motor cortex dies, then it's not sending that information to the basal ganglia. And therefore, the basal ganglia should be then, which responds to the primary motor cortex inputs, should be behaving differently. And therefore, you should not only have a difficulty in performing the task, but also have changes in the vigor with which you perform the task, or how it relates, or performing the tasks as it relates to your motivation, which we know are functions of the basal ganglia. And this paper set out to understand how basal ganglia and striatal activity changes after the stroke, and whether that's correlated with the recovery or the kind of the transfer of function to the perilesional cortex from the primary motor cortex. Okay, cool. So how do you do this in mice? In rats. So what they do is they have this task, which I think is like really crazy. So in response to a reward, the rat has to reach through a hole with its upper limb, and then manipulate a lever by grasping it and kind of pulling it. Tim's giving me a face. It's not a lever. It's
is that what it is?
It's a reach to grasp, I thought.
Oh, I thought they just grabbed I just thought they grabbed the pellet itself.
Oh, I thought they had to grab something. There was some other instrument.
I didn't read it super duper closely, so please continue. I'm going to search through it.
No, I could also be wrong.
But continue.
Okay, so they reach through. So there's two components of the motor action, right? So there's the gross motor action of reaching into the hole, and then there's the fine activity where they can actually manipulate their paw to either grab the pellet or grab something else.
I looked it up. It is a pellet. So they have to basically, there's a tiny little slot, and they have to reach with a paw through the slot to grab the pellet, and then bring the pellet back. It's a bit tricky so that they grab the thing that they're trying to get, as opposed to grabbing a joystick. It's probably going to be similar, but the difference with joystick is you don't have to hold the joystick back, right? But if you have to grab the pellet, you have to bring it back. So It's kind of like, when a piece of chocolate that you're eating rolled underneath your couch, and you're trying to fish it out so that you can eat it before the 5-second rule. So it's a bit like that for the mouse, for the rat.
And I think that that's actually a great analogy, right? Like, so you'd expect that someone with a stroke might not be able to reach into the couch and get the chocolate, but if they had basal ganglia dysfunction, they might not be able to do it fast enough to get it within the 5-second rule. So they might be able to perform the task slower, but they won't have the vigor or the motivation to accomplish the task that quickly. So what they do is, before the stroke, they train these rats to kind of peak performance in grabbing their food reward. And essentially what we know from motor learning studies from this group and others is that sorry, I keep saying mice, but rats will kind of...
It's the same. It's the same to us.
Yeah.
We won't be offended.
I think
the rats will be offended.
I think good behavioralists might be offended, but they the rats will reach in and they kind of perform this stereotyped activity. So animals will have kind of variability in what their like movement paradigm is, but they will perform that set of actions in exactly the same way every time after they're trained really well on the task. And this is work from this guy, Bence Olveski, I think is how you pronounce his name. Where, I mean, these are just like phenomenal videos. I think Stefan Wolf is the first author, where they essentially train these rats on this motor learning task. And after they get overtrained, essentially, like, you see some rats where, like, in order to get itself to start like the sequence to accomplish this motor task, they'll like do a loop-de-loop and then jump through a hoop. And then, I'm exaggerating, but like that rat, like, for whatever reason, it was exploring the action space. It did this thing, got the reward, and then it just like kept doing it because that's what it like learned to do to get the reward. So after they give the stroke in these rats, what happens, right? So what's interesting is that there are a number of different parameters of the movement that they can look at. So first of all, you could imagine that they might get fewer pellets, and they do get fewer pellets. But they also have, when they get the pellets, they do it with a lower speed, and there's a high variability in the speed with which they get the pellets. So many different parameters. So I think that, certainly successful trials go down, but also on the successful trials, how they accomplish the task also goes down. And I think that that's really an interesting piece of that.
Yeah, the authors plotted the trajectory of the rat's paw as it reaches through the slot to try to get the pellet. And before the stroke, it is all very consistent. Everything is like very, very tight. But after the stroke, the rat is like kind of, you see like a lot of divergence, and the rat's hand is like going all over the place.
I think the visualization of this paper is really nice.
Yeah. But Ori, you're not going to talk about how they did the stroke, because I was insane. Because
I wasn't going to talk about it, but you guys should talk about it.
Oh, okay. So like normally, like when you have a stroke in humans, you have a blood clot and it obstructs a capillary or whatever blood vessel in the brain, and then your brain doesn't have oxygen, you die. Well, the brain dies. I think in animal models, what people sometimes do is they open up the brain, they find a blood vessel that they want to cause a stroke in, and they tie a knot to block the blood flow, and then neurons around that place would die. But in this paper, but the downside is you often cannot exactly control how big the spread of the stroke is. So the region that dies might be quite variable. For this paper, they want to make sure the stroke happens in the motor cortex. And what they did is completely weird. They injected a dye into the vein of the rats, and this dye is somehow photosensitive. And if you shine the light onto the rat's brain as this dye is circulating, then the area right underneath the light, those dye would react with the light and then like cause massive local damage by reacting, like it causes cell death of the blood vessel wall, and that causes blood clot just within that region. And I didn't realize that dye, like just like it's called I can't remember what it's called. It's a red dye.
Bengal something.
Yeah.
Probably Bengal red.
Yeah.
Something like that.
Rose or Rosé Bengal. No, Rose Bengal.
Is it rosé? Is
No,
it
rosé.
is it
It
from
does
is
not
it
have.
drinkable?
what's on
I
my
don't think
mind
it
at-
has that. I don't think it has an accent.
at 10:40 in the morning here. Yeah.
But yeah, that's I didn't know you can cause damage to your brain by like shining a light on a dye. Completely new to me. But anyway, sorry. Great
no,
digression.
but I think that what's cool is that this is a way for them to get a really targeted area of a lesion, right? And what they do in the methods is they actually put like aluminum foil around everywhere but the area where they want to illuminate, and then they can just get a very targeted lesion. And this, as I said before, really allows them to understand the infarct, control the infarct size, which then allows them to have less heterogeneity and kind of recovery trajectories essentially after the stroke. And then also obviously limits their generalizability to human stroke. But, in these mechanistic studies, this is an important piece. So what they then do, and I think that this is a really important piece of kind of clinical management of stroke patients, is that you send them to rehab, right? So they, if you repeat a motor action after a stroke, you can get better at it and relearn. And Tim has done this in the context of dopamine lesions in unpublished papers. But in this case, what they do is they, or maybe recently published, but
don't go there. Please don't go there.
So, but what they do in this case is they then take the rats, and so you have pre-stroke, you have how they do in their motor function. You give them the stroke, then the next day or the next session is their immediate post-stroke outcome. And then if you give them the same session over 8 days, I think they do it, what you can see is that they slowly will recover their motor function, and they improve in the kind of the number of correct trials, the number of pellets they get. They improve in their speed, and importantly, they improve in their consistency. So the variability of how they behave goes down. And that allows us now to say, okay, well, if the rats get better at this motor task over time, can we record from different parts of the brain during that recovery period and say what neural activity changes correlate with recovery of function? And that's what they do. And they put probes both in the perilesional cortex, so in the area around the stroke, which we know from other papers then takes over a lot of the motor cortex activity. And they put probes into the striatum. And they put it into a part of the striatum that is kind of more maybe motor focused and less cognitively focused, called the dorsolateral striatum, that receives inputs from primary motor cortex or the perilesional cortex. And what they can do is they can record the activity of the striatal neurons and the cortical neurons and say, for example, one is what happens right after the stroke in the striatum. So does the activity change, or is the activity intrinsically generated and does it stay the same? Does it improve? Does it change? Does it go back to normal if it changed initially with motor recovery? And then also, how does kind of the time locking or variability change between the perilesional cortex and the dorsolateral striatum? And could that predict recovery of this motor function? And before we get there, I think one important thing that they do is an intervention experiment, which I was buried in the supplement, but I think is actually like a super important kind of point to get at their hypothesis, although they try to get around this computationally. So I think the crux of their question is the striatum important for motor recovery after stroke? And by recording striatal activity, you don't get at that question, right? You could say, well, striatal activity is associated with motor recovery, or coherence between motor cortex activity and striatal activity is associated. But what if we inactivate the striatum? Does that reduce motor recovery? And they don't quite do that experiment, right? But what they do is they give an inhibitor of activity called muscimol, which potentiates GABAergic inhibition in the striatum. And what they show is they reduce the kind of the motor function, or they make the rats worse at this task after the stroke. We can debate about these. These are kind of very, very complicated experiments, but I think what they're saying is that they know from previous work that this task is dependent on striatal activity. And with this experiment, what they're saying is that after the stroke, this task is still dependent on striatal activity. Yeah. what would have been really, really cool would have been to essentially inactivate these neurons over, not during the task itself, but for example, like you throw, so I guess take a step back, we think about motor learning happening in different phases. So, the brain is important for the performance of the action, but also the brain is important for learning the task so that they get better for the next action. So you could have imagined an experiment where they actually, threw the rat into the chamber, give after the stroke, they perform the action, then they take them out, and a lot of learning is consolidated overnight. They could have inactivated the striatum overnight and then put them back in the next day and said, oh, is the striatum important for the recovery from the relearning, or is the striatum just important for performance of the action? And they didn't dissociate those two.
I don't even know if that is known for non-stroke motor learning. Like if an animal is learning to reach for a pellet, because even in a non-stroke rat, you put the rat in a box and it has to reach to grab this pellet. Even a normal rat would have to get a bit of learning to get this going. And I don't know whether they have looked at consolidation in the striatum.
I think they have for like rotor rod tasks for those. There's definitely consolidation in that striatal dependent.
So I'm curious about the consolidation overnight thing. I don't know anything about how that is studied, but mice are nocturnal, right? Is that true?
Sorry, I guess I mean over their sleep period. So
but they don't go to bed and then wake up 8 hours later like a human, right? It's kind of intermittent, especially because we're coming in and out of the rooms and doing things with them. So
yeah, I think the way,
how do you measure that?
I mean, they definitely sleep, but I think the way you do it is by doing the experiment where you inhibit something after the task.
But actually, related to this, it is known that, for example, in songbirds, like zebra finches, these are birds that like to sing songs, and they learn it from a tutor bird. So a tutor would sing a song, and these baby birds would try to learn to sing it. And I know this because Mike Long at NYU does a lot of these experiments, and his group presents every now and then, and it's fantastic data. These birds, if you record from the striatum and also from the version of the cortex in these birds, you can record the pattern of neuronal firing when the birds are actively learning these songs. And you see a pattern of firing, and then you let these birds sleep. And when they're sleeping, you see the same pattern being replayed. And the hypothesis is that these replaying when the birds are sleeping or when the birds are not actively singing is important for the consolidation. And I know less about non-bird, like in rodents learning. I actually know a lot less because that person is not at NYU.
No, I think that's definitely known that the striatum is important for motor learning, like consolidation of motor learning.
But for replay.
I don't know about replay. Yeah. Sorry, I don't know about replay. And I think so. So I think that those are kind of open questions, and I think that they allude to that in the discussion. They talk about does the dopaminergic production to the striatum play a role in stroke recovery, and these are things that I'm sure they'll work on in the future. But so what are their findings, right? And I'm going to give an overview of what I took away from the paper, which is dense in the maths, as Tim likes to say. But so first of all, in the striatum, the activity is decreased during both at baseline and during the task performance after the stroke. So we know that this is very well known that striatal activity is highly dependent on the excitatory inputs from the cortex and the thalamus. Essentially, these neurons are like quiescent, except for when they receive excitatory inputs. So as you could imagine, if excitatory inputs from the cortex go down, their activity would go down, and that's what they saw. And that the other interesting thing is that recovers with retraining, so correlates again with the motor recovery. And they also show that there's a decrease in the variability of their firing with recovery. And then they look at the next piece that they look at is what kind of the parameters of the motor task the striatal the striatal activity correlates with. So, for example, I said it could be, they measured the success rate, they measured the speed of their grasp, they measured the variability of the speed of the grasp. So if you would expect the striatum to be involved in motor recovery after this stroke, you would expect that it would be modulating one of the outcomes that we know the striatum is involved in. So, for example, vigor, speed, or variability. And in fact, that's what they see, that the recovery of striatal activity correlates most strongly with the grasp speed as opposed to other parameters of the movement. And then the last thing is that if they do these dual recordings from the perilegional cortex or the striatum, that the activity is highly correlated, and the recovery of activity in the perilegional cortex is highly correlated with recovery activity in the striatum. And so these are kind of roundabout ways of saying that very clearly that they that these two theoretically should be kind of in partnership during the motor recovery period. And then there is a black box of math that I cannot explain, where they essentially, it's very cool that what they essentially do is they say, based on these neural activity parameters, can we predict the recovery trajectory of an animal? And they can, and including the striatal activity in that predictor is makes it a better predictor. And I think that is kind of a very interesting piece, because it's really, it explains an additional piece of how we could say, okay, person A has a stroke, person A is going to recover to X level of their original function. Maybe that would be better if we could predict that better if we had some information about how the striatum is working. Tim, I don't know if you want to add any more about the maths part of that.
Absolutely not the maths, because I don't think I can explain it. Well, I didn't understand it too well, the canonical correlation analysis, because I didn't quite have time to get into it. But I thought, can I just re-summarize what you said and
Sure.
correct me if I'm wrong, just so that I make sure I understand it, which is these researchers found that after the stroke, on the first session of rehab, we put the rat back in, and the rat moves erratically when it's trying to reach for the pellet. So the trajectory of the paw is all over the place. And at the same time, they recorded from the striatum, and they found that even though the rat is actually making some movement, but during those movements, fewer cells are recruited in the striatum.
Correct.
And those fewer cells that are recruited, they are also firing very erratically as well. They don't fire stereotypically. They're it's like from trial 1, this cells would turn on at this time, another cells would turn on at a completely different time. And trial 2, those cells would fire at a completely different place. So there's no consistency at all in the brain, and also in the mouse's paw movement. And interestingly, the same for the perilegional cortex, which is the secondary motor cortex, also shows this kind of behavior, this kind of changes, which is quite odd. Like, for example, I think they found that in the perilegional cortex, so secondary motor cortex, stroke in M1, the primary motor cortex, leads to fewer cells in the secondary motor cortex to get recruited during the paw reach movement. So like the entire brain shifted, basically. Well, not the entire brain, 2 regions, both shifted when the primary motor cortex changed, which tells you a lot about how the whole brain is like. There's a lot of connection between these different regions. But like after the mouse as the mouse is showing improvement. Sorry, I'm doing the same thing. I'm sorry to my rat and mice and human friends. As the rat is going through rehab and reaching more consistently, more cells get recruited in both regions. And not just that, these cells are firing more and more in a stereotyped pattern as the behavior is becoming more and more stereotyped. Is that correct?
Yes.
So it's looking more and more like the pre-stroke rat behavior. Like the brain is firing in a way that looks more and more like the pre-stroke brain, but in some subtle sense, which is subtle differences, which is the cortex that is recruited is different because the original bit of cortex is now gone. And what's actually quite interesting is that if you look at the striatum, they showed it in one of the supplemental figures. Even inside the striatum, so when they're recording for the striatum, they have different probes at slightly different location of the striatum.
Oh, yeah.
And what they found is that even within the striatum, the area of striatum that gets recruited also changes as the mouse goes through rehab. So it suggests that the whole brain really is reconfiguring to this.
Yeah, I mean, the data is so dense that they get out of each one of these rats, right? I mean, it's awesome. So yeah, I think that kind of wraps up the points from this paper. I think that, to me, in some ways, like, it's such a straightforward question that they ask that it's like, and they do it in a very elegant, they answer the question in a very elegant way. And they certainly raise many, many, many more questions to go forward. For example, are there neuromodulators that are important in the striatum? Is the striatum important for reconfiguring the motor cortex, or is it kind of just supporting the motor cortex actions or a recovery, et cetera?
Oh,
yeah, go ahead.
Although it's probably quite important to point out that these rats, even though the brain has remapped to allow these rats to go through to recover, they don't quite recover to the before stroke level. So there's still some impairment left. So it's probably quite important to point that out, that your brain isn't infinitely plastic, which it's quite nice tie into a while ago. I think I presented a bunch of papers about you can rewire ferrets at birth, that if you take out the ferret's visual cortex and rewire the auditory input rewire the eyes to the auditory cortex, the ferret would just learn to use the auditory cortex to process visual information. So in a way, this is kind of a slight parallel to that, in that you destroy the primary motor cortex, and the secondary motor cortex kind of takes over, but it doesn't quite do it 100%. As you can imagine, the brain isn't like, you can't just do this and take chunks out and the brain would fully readapt.
And then I just was going to add one more thing, which was that, if we understand the neural activity patterns, you could imagine that that you could stimulate non-invasively, for example, with like transcranial magnetic stimulation, or you could have brain-machine interfaces that could recapitulate this activity and improve function. And then also the other thing is, like, you could decode the neural activity and have a robotic arm, for example, perform the task. And I think that, there's certainly work happening here at UCSF by neurosurgeons that are taking kind of brain-machine interfaces and producing speech from activity in primary motor cortex in patients who can't talk. And I think that in some ways that's the way of the future, but probably a bit ways off.
I have a question sorry, a bit of a tangent. I have a question still regarding the assumption of this paper, which is, if you take it somewhat at face value, that before stroke, your primary motor cortex is the brain region that is doing all responsible for the grasping and reaching for the pellet, and then after the stroke, this gets kind of farmed out to the secondary motor cortex. Then would you expect, if you were to inactivate the secondary motor cortex before stroke, it doesn't do very much. It affects the rats one way.
Like during training.
But after stroke, sorry?
Like, you're saying inactivate the secondary motor cortex during training for the task.
No, just look at, like, before stroke, you inactivate the secondary motor cortex. It might have some effect on how the rat reaches and grasps. But after the stroke and after rehab, when the secondary motor cortex now becomes a bit like the primary motor cortex, then inactivating this region would give you a completely different behavior phenotype. That would look a bit like what happens if you were to inactivate the primary motor cortex before the stroke. If you see what I mean.
I was going to say something even different, which is that what if you inactivated the secondary motor cortex during training, and then you give the stroke? Then does the secondary motor cortex not take over the function of the primary motor cortex?
It will be the tertiary motor cortex.
Yeah, exactly.
that they discover.
Yeah. Cool. So yeah, that was a fun paper. I think it allowed us to talk about stroke, a lot of motor learning, and some scary math. So we can wrap up unless anyone has other thoughts.
I actually do have one thing. It's tying back to your original question about neglect. So if you have a stroke just in the primary motor cortex, like for example, for these rats, they can relearn as shown by this paper, even though not 100%, but they can still grasp. At the end, they can grasp it. It's quite reliable. But Ori, you mentioned if these rats were to have neglect, then they never relearn.
Well, I think they relearn worse. Yeah, they don't relearn as well.
Okay. So yeah, I'm just wondering how. I guess we don't know how neglect works, whether it's cortical mediated or whether it's subcortical striatum mediated.
Certainly cortical mediated. I think the question is like, do they not engage in rehab and therefore they don't have recovery, like they don't have improvement in the motor cortex because they're inattentive to that hand? Or like, is there a true like neural basis for like some sort of inhibition of the motor cortex recovery, right? Like if you could force them.
I see what you mean.
So like there are some people that there's like all these kinds of really cool like physical therapy and occupational therapy paradigms where, for example, like if you have neglect of the right hand, you can actually like move the right hand to the left side of the visual field and you're like focus on the right hand. This is your right hand. And then they will actually do better than if, for example, you have it on the side that they're neglecting. Or
that's kind of like mirror therapy for individuals who have had a limb amputated and they would have pain in their limb, which is no longer there. But if you put a mirror, so I don't know how this works, but put a mirror so that they can they use the reflection of their left hand to kind of fill the space, as it were, that their right hand would have occupied, and this can ease the pain. And I don't know to what or like how it helps rewire things, because like you were mentioning before, that the kind of what M1 does either kind of gets sent out into the surrounding areas, but then also there was probably a sprouting of M2 into the M1 area. But the same thing happens in the somatosensory cortex when a limb has been removed. You have, and that it's a little bit different because then it sounds like that with the motor cortex, you don't have like kind of extraneous movement in other regions that, would have been represented in the M2 prior to the stroke. But with respect to the lesions in this or loss of a limb, the regions actually like come in and take over. So then take over the region that was once relevant for the right hand. And then you get referred sensation. But in any case, I don't know if the mirror therapy actually helps to like get rid of the referred pain or the limb, the pain associated with the phantom limb. But I think, like that would probably be an easy, or a well-known, it's already used a lot, so it probably would be helpful.
Yeah, I think that like those kinds of paradigms are so cool, and like they're not invasive. You're just like they're intense economically for the system because you have to have like intense therapy, but they're just like fantastic, low risk, high reward interventions.
I wonder how that works, like the phantom limb and the mirror. I mean, it makes like presumably you are rewiring the brain as you put a mirror in and getting the correct visual input to the missing limb. But
it doesn't like bring that limb back. Right.
No, it just makes the pain go away. And then
right. But can you can you over time get less and less pain by doing? Because if you can reduce the pain over time by just engaging in this therapy frequently, then it really does seem like this actually is rewiring things. Just simply imagining that your limb is present can change, change connectivity and
you're fooling your senses in a good way.
And it's strange because it's like the neurons that would convey that nociception, like they're like they're not in the brain. At least the primary neurons.
Well, I think that's like centrally mediated pain, like, right?
And it's completely wide open. No one knows what pain is inside the brain, right? As far as I can tell. Like there's no pain. There's no like a well-defined pain region in the brain, right?
or necessarily, or like we talk about glutamate and GABA, but
Oh, there's no pain. There's probably not a pain neurotransmitter. Oh, there is
I don't
one.
know
It's called
if it's
capsaicin.
Right. Well, that's peripheral, right? And like there's a cocktail of things that we think lead to pain, but I don't know anything about it in the central nervous system. Maybe my comment is not even relevant about like, is there a neurotransmitter or neuropeptide or something like that tends more to drive pain sensation? Probably not, but I don't know.
Oh, well, there is. So there's opiates. So that's one. That's that's a
right.
big
That's
one.
true. That's true.
Yeah, but I think the neuromechanism. Maybe one day we'll talk about pain on the podcast, and we'll have to dig into some paper. But I think, as far as I can tell, it's completely wide open. We don't know if there's a single brain region. Is it a certain pattern of firing across brain regions? Who knows?
We should definitely do that. I bet there are a lot of people out there who either have personal experience or know somebody who,
Should we wrap up?
Mm-hmm.
Okay. So TWiN show notes will be at microbe.tv/TWiN. Vincent will get those up when he posts the episode. We definitely love to get your questions and comments. Send them to twin@microbe.tv. And then consider supporting the science shows, both TWiN, but also TWiV, TWIM, Immune, many of Vincent's other shows at microbe.tv/contribute. Thanks everybody for joining us. This was fun. It was nice to talk about science after 6 weeks of just talking about breast milk.
And rapidly decreasing brain function from all of this.
Right.
Exactly. So I'm Ori Lieberman. I'm at UCSF in the Department of Neurology. You can find me at Ori Lieberman on Twitter. Thanks, Tim, for joining us. Tim Chung is at NYU.
Thank you. Thanks, Ori. Thanks for being
and
nice paper, Ori.
Thank you. And then Vivian Morrison is now at Tulane. I need to change that in the show notes. And if you'd like to connect with her, you can find her on LinkedIn. And we've missed Vincent and Jason. They'll be back next time. You've been listening to This Week in Neuroscience. Thanks for joining us. We'll be back next month.